The Cancer Genome Atlas, CGA, was a comprehensive research initiative aimed at understanding the genetic basis of cancer. It was a collaborative effort between the National Cancer Institute, NC, and the National Human Genome Research Institute, NURI. CGA's primary goal: to analyze and characterize the genomic alterations in a wide variety of cancer types to provide insights into the underlying mechanisms of cancer development, progression, and treatment. Key objectives of the CGA project included: comprehensive genomic analysis. CGA aimed analyses of various cancer types to identify genetic mutations, copy number variations, epigenetic changes, and other molecular alterations that contribute to cancer. Large-scale data collection. The project involved collecting a massive amount of data from thousands of tumor samples, including DNA sequencing, RNA sequencing, epigenetic profiling, and clinical information about the patients. Data sharing. CGA emphasized the importance of making its data publicly available. This enabled researchers worldwide to access and analyze the information, accelerating discoveries and collaborations in cancer research. Biomarker identification. By identifying genetic and molecular changes associated with different cancer types, CGA aimed to discover potential biomarkers for early diagnosis, prognosis, and treatment response prediction. Personalized medicine. CGA's insights were expected to contribute to the development of targeted therapies and personalized treatment approaches based on the specific genetic characteristics of each patient's cancer. CGA focused on a wide range of cancer types, including but not limited to breast, lung, colon, ovarian, and brain cancers. The project involved a multidisciplinary approach, integrating genomics, bioinformatics, and clinical expertise. The CGA project officially launched in 2005 and completed its major data collection efforts in 2016. The generated data has had a significant impact on cancer research, leading to numerous scientific publications and insights into the genetic underpinnings of various cancers. Analyzing data from the Cancer Genome Atlas, CGA, involves processing and interpreting the vast amount of genomic, transcriptomic, epigenomic, and clinical data collected from various cancer types. This analysis can provide insights into the genetic alterations, molecular mechanisms, and potential therapeutic targets associated with different cancers. Here's an overview of the steps involved in CGA data analysis. Data access and preprocessing. Obtain access to CGA data through authorized channels or repositories. Download relevant data files, which may include raw sequencing data, FAST files, process data, BAM files, clinical metadata, and annotations. Preprocess the data by aligning sequencing reads, correcting for technical artifacts, and filtering out poor quality samples. Genomic data analysis. Identify genetic mutations, single nucleotide variations, insertions, deletions, and structural variants, gene fusions, copy number variations, using variant calling tools. Perform somatic mutation analysis to distinguish between germline and tumor-specific mutations. Analyze mutational signatures to understand the underlying causes of DNA damage and repair processes in cancer. Transcriptomic data analysis. Quantify gene expression levels using RNA sequencing data, RNA-seq, and identify differentially expressed genes between tumor and normal samples. Perform pathway enrichment analysis to understand biological pathways and processes that are dysregulated in cancer. Explore alternative splicing events and isoform expression patterns. Epigenomic data analysis. Analyze DNA methylation patterns using methylation array or bisulfite sequencing data to identify differentially methylated regions. Investigate histone modifications and chromatin accessibility to understand how epigenetic changes impact gene regulation in cancer. Integrative analysis. Integrate genomic, transcriptomic, and epigenomic data to identify potential driver genes and pathways associated with cancer development and progression. Perform integrative clustering to stratify patients into molecular subtypes with distinct clinical characteristics. Clinical correlation and survival analysis. Correlate molecular findings with clinical features such as patient demographics, tumor stage, and treatment response. Perform survival analysis to assess the impact of genetic alterations on patient outcomes. Biomarker discovery and therapeutic targets. Identify potential biomarkers for early diagnosis, prognosis, and prediction of treatment response. Prioritize genes and pathways that could serve as therapeutic targets for developing precision medicine approaches. Data visualization and interpretation. Generate plots, heat maps, pathway diagrams, and other visualizations to represent the findings effectively. Interpret the results in the context of existing literature and biological knowledge. Validation and follow-up studies. Validate key findings using independent datasets or experimental techniques. Perform functional studies, in vitro or in vivo experiments, to confirm the functional significance of identified genetic alterations. Data analysis requires expertise in genomics, bioinformatics, and cancer biology. Researchers often use a combination of programming languages, such as Python, R, and specialized software tools for specific analyses. Collaboration between computational biologists, clinicians, and experimental researchers is crucial for translating key insights into improved cancer diagnosis and treatment. Some of the primary resources for data analysis include Genomic Data Commons, GDC. The Genomic Data Commons is the official data portal for data. It provides access to raw sequencing data, process data, and clinical information. The GDC Data Portal offers tools for data exploration, visualization, and analysis. Researchers can use the GDC Data Transfer tool to download specific datasets or portions of data.